Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome back, everyone, to the Genetics Podcast. I'm excited to be here today with Ness Bermingham, who's a scientist-turned-biotech entrepreneur who's really dedicated his career to using some cutting-edge science to solve big healthcare problems. He's co-founder of Intellia Therapeutics, Korro Bio, a number of companies. I think we're up to 6 biotechs now, and counting probably. But he's an operating partner at Khosla Ventures, chair of the board at Korro Bio, and it has just an incredible amount of experience in this space. So is it 6 or 7, or what are we up to right now in terms of?
Let's just say it's north of 6 for sure.
Great.
We've got to include the failures also in there.
Yeah. So when did you decide that you wanted to start a company? What was the first one? And take us back to that story, why you decided to go down this road.
Well, so first of all, thanks a lot for inviting me to be on the podcast. I love to talk about biotech and hear other people's views, so I really appreciate the opportunity. I sort of fell into it really on the operating side. So I was very fortunate in getting into venture capital at the end of 2001, early 2002, joining Atlas Venture, coming out of Wall Street, where I worked in equity research. And it was, it really started, when I first started, it was very much about the sort of the financial aspects of building a company, right? And sort of the conceptual components of what a drug is, how to build a drug and develop it. And the practical aspects of culture, people would talk about things like culture. You never really knew what that was until actually you were steeped in an operating role and actually understood culture on a day-to-day basis within an organization. And I remember being absolutely offended when a senior leader in a biotech company made a general comment about VCs, had the vast majority of us had no operational experience. So we pound the table, every quarter at the board meetings, but we didn't know what it actually meant to execute on that and realize that within an organization. And I remember being offended by it. And many years later, I still remember who said it actually, but many years later, I ended up in an operating role by default and realized actually how, prescient that actually was, that the reality was building these companies when you're actually rolling your sleeves up and doing it, as many of your listeners know, it's actually not that easy. And when you look at the overlay, as you and I talked a little bit about, between what's happening from a macro financial standpoint versus the fundamentals of your company, really actually starting to see the linkage between those two is also critical. So first company was a company we called Telemedical with PureTech, and Steve Paul. And that really was in the major depressive disorder space and PTSD. And that was a fascinating company. It was a device. And that really was my first sort of foray into it. But clearly the biggest one, then really came into Intellia. I built a couple of others, but then Intellia really was the one that sort of took off. And we moved really rapidly, less than 2 years from concept, piece of paper. I think it was 250K I got from Atlas to start the company into IPO in less than 2 years. It was an unbelievable ride.
Wow. What was it that drove the speed of that? You talked about macro being a factor. Was that a great time to start and scale biotech companies, or had you hit lightning in a bottle technology-wise, or combination of the two?
It really was a combination of the two. I mean, lightning in the bottle would be CRISPR-Cas9, the idea of genome editing. People had talked about it for a long time, but there was a reason that CRISPR-Cas9 really caught people's attention. So you literally Thanksgiving, you're around the Thanksgiving table, and people would actually start talking about CRISPR-Cas9 and actually start talking
Yes.
about genome editing, which is really phenomenal. I think there also was this convergence of what was happening within biotech. I've said a number of times, if CRISPR-Cas9 had been identified, 15 years earlier or 10 years earlier, I don't think we would have seen it advance the pace that we did. So that convergence really was around things like
Mm.
whole genome sequencing, population studies, novel pathway identification or targets for disease intervention. The work that people or companies like Alnylam and Ionis actually did as we look at delivering nucleic acid to cells, how to actually do that, how to manufacture it, all of that stuff. I think CRISPR-Cas9 happened to come at a point where there was such a level of data that was out there and sophistication that allowed us then to move it forward extremely rapidly. And clearly, first drug already approved in that space, I think, which is almost unprecedented for a novel modality.
Yeah. Yeah. How did you feel when you saw that news? That was pretty amazing.
It was absolutely phenomenal. I was talking to Matt Porteous, so Matt and I originally were working with CRISPR Therapeutics, so with Roger and with Sean. So, we sat in the room, we were talking about the application sickle cell disease. So to see that move all the way through and actually, help patients, is really phenomenal. I mean, the speed at which it was developed, and I think CRISPR and Vertex have done a phenomenal job, in really focusing down and getting to approval, getting it reimbursed, and now getting it into patients. And we're starting to see the real impact on that. So I think really that is amazing. I think the work that Intellia, we took a very different strategy when I built Intellia was let's deliver the drug directly into the body and edit cells in the body. And looking at the data that's coming out from that, really, again, it's just been, beyond expectations. We talked about one and done sort of treatment paradigms, inject you or deliver to you once, and then you have an effective curative effect. Seeing that actually play out in reality is really pretty spectacular, and seeing it across more than one indication. So I think that it is pretty, it's great how we've seen that transform actually now into therapeutics. But I think it's important to remember, anybody could have done this. There's this thing about, well, did you have a vision? Was there something, that you had when you started these companies? And I think these technologies, the foundational data that was already out there, the work that had been done, I think a lot of people were actually looking at the application of these technologies. And really, we had three companies that moved very rapidly in
Mm.
almost in tandem: Editas, CRISPR Therapeutics, and Intellia, that really then allowed us to be able to move forward quickly.
Yeah, interesting. I was going to ask about Korro Bio as well. I'd love if you could talk a little bit about that, but it strikes me that it's a similar pattern. It's RNA editing, and not there's not 100 of these companies, but there's also not just one, right? And so it's an interesting dynamic where people are kind of coalescing around a new technology around the same time. I'd love if you could talk a little bit about that because that's another wave that feels exciting to me, at least that's happening right now.
It's so, it is interesting what we've seen around the number of companies that now that we're seeing within biotech being founded, and many of them coalescing around singular technologies. I'm sure we'll get into AI at some point and what we're seeing within
Yes.
that space. So if you look back historically, within, for example, siRNA, you'd one or two companies in the space. Antisense, really, you'd only one. Isis Pharmaceuticals, which is now Ionis. Within gene therapy, you kind of had a very limited number of companies in space. So now we're seeing this actual rapid explosion almost around technologies and multiple companies. And then you get into the differentiation of them. So, I think when I look at biotech today, there's a tremendous level of noise that's out there, and the differentiation factor becomes more and more challenging for them to be heard and actually
Mm.
garner investment. Um, so Korro is an interesting one. So when we looked at Intellia, going in and modifying DNA, that's a permanent modification that you're obviously doing. And the challenge I always had with Intellia was I never wanted to go in and do a modification to a target that I didn't have a lot of data to validate it as a target, but B, from a safety standpoint.
Yeah.
So when you look at, the utilization of things like siRNA and ASOs, there was a tremendous number of targets they went after, a lot of data around it. So we effectively were very comfortable when we went after things like ATTR
Yeah.
and HAE as an example, right? For Korro, one of the areas really we were very interested in was, well, is there a way to actually co-opt an endogenous-based system that edits your RNA, and do it so that we can do it in a way that is not actually permanent, right? So we can
Mm.
titrate. We can switch it on, switch it off, reset a cell if we needed to. So give us a much broader applicability than what CRISPR-Cas9 really is good at: switch stuff on, switch stuff off, like effectively switch
Yeah.
off permanently, right? So now, easier said than done. We now had to learn about how to recruit this thing called ADAR that sits within our cells and actually, edits your RNA at specific sites. So fascinating that there's an endogenous system to edit RNA there.
Right.
So I think there were probably two key questions when we started the company. Question one was, how do you really co-opt it to target the site you want it to actually target in a construct, an oligonucleotide that is therapeutically and commercially viable, right?
Yeah.
And then the second one is, where do you deploy it? So, it's I believe that any modality we look at, it has to do something that's better or different than other modalities out there that it is specifically applicable to, that the others
Right.
just can't do, right? I think we're seeing a lot of various mouse traps that they catch mice. One might be gold, one might be silver, one might be a little
Right.
bigger than the other one, but frankly, they all do the same thing. So it's, how do
Yeah.
you actually differentiate yourself? And we spent, I would say, a lot of time within Korro, looking at that and trying to understand what is the best application for this modality versus other modalities that are out there. So, that's how we've built the company. And as they continue to roll out their pipeline, I think you'll see the actual applicability of recruitment and utilization of ADAR. What has been great is we've already seen translation now into the clinic from other data that's out there. And I think that that's looked positive
Yeah.
from the fact of, yes, in a human, you can deliver, you can recruit ADAR, and you can modify RNA. And now the question comes out about potency of these molecules, moving outside of the liver, and then the actual target, what you actually want to do with that protein or with that RNA target.
Yeah, I think alpha one antitrypsin is one of the targets there. I would be interested in your perspective on that as a therapy area. A lot of people are going after it with lots of different modalities. Is that because it kind of hits this sweet spot of, we can deliver to the liver? It's common enough that it's quite commercially viable, but it's also, I guess, rare enough and genetically driven enough that you can go after it. Is it in a little bit of a Goldilocks zone, or what is it about that disease that makes it so hot for precision medicine companies?
So yes, it is in the Goldilocks zone. And what's interesting about it is you've got two issues. You've got lung and you've got liver issue, right? So the
Yeah.
liver secretes it and it goes to the lung where it's active. So you have to deal with two separate, effectively, issues in the body when you actually want to treat this disease. Most approaches will deal with one of the two, right? You either deal with the liver or will deal with the lung, but it tends not to deal with both. So I think that that's one of the unique things that Korro is going after, that really lends itself uniquely to this modality. So that's the first thing. I
Oh,
think the
interesting. Lung
second,
and liver at the same
that
at the
you can
same.
get both, right?
Yeah.
So you can deal with the actual fundamental genetic driver, for the Z
Yeah.
allele, but actually then you can, through that targeting, you can deal with the liver pathology, and you can deal with the lung, and
Yeah.
that's absolutely critical that you do both. The second aspect around it is really around, as you look at biomarkers. So I'm a big believer also that first indication you go into, that you limit the uncertainty that you have. So to your point about delivering to the liver, that's taken off the table for you,
Yeah.
right? So, limit as much as you can the uncertainty around the indication, so that the data, when it comes out, you can actually interpret that data. Because in many cases, it's never black or white. It sort of sits somewhere between it. So that really
Right.
you can interpret it and target it down to your modality, especially when it's your first indication.
Mm.
And then the second one is that you've got markers that you can actually use from a pharmacodynamic standpoint, that is telling you from that dosing paradigm. So you can go in with, based on your non-human primate studies, you can go in with a plan on how you want to dose and your phase one, but really getting that biomarker data out really then starts to tell you realistically in a human what's actually going on, how often you need to dose, how to actually think about that. So I like indications too, and alpha-1 antitrypsin is a great one for that, because you're getting this, this protein released from the liver, you can actually detect it, look at it, and actually show the implications of it. So that also makes it a great indication. And then from an FDA standpoint, you've got a clear clinical development strategy as you think about how to actually move it forward and get approval for it and ultimately leave into commercialization.
What is you mentioned earlier actually that person who said that investors or probably more generally people who sit outside of early stage venture-backed biotechs don't always understand how difficult it is. I'm curious to just get your perspective on what you've learned over your career. What is it within that? Is it weathering the ebbs and flows of the public markets? Is it about actually building a small team that has the right culture that can do something really difficult and working together through those challenges? What is it that people don't understand if they haven't been in that driver's seat for a couple of times?
I think that the biggest one for a lot of CEOs that are first-time CEOs today is the fact we're going through this massive cycle right now, right?
Yeah.
Where you went through where there was a tremendous volume of capital that was flowing into the sector. A lot of companies were able to go public. And now we're in a really capital constrained environment, where the cost of capital is very high. Um, I think there's a couple of aspects as we think about, building our companies and the considerations for it. So one is, before you really get clinical data, the fundamentals of the company are very important. Differentiation in the marketplace, being able to explain that and communicate that easily, that people actually understand it. Um, but there's also this sort of macro or technical component, right? That the fundamentals of you at times don't really matter, right? And we kind of saw this with triplet, that what was going on within the disease space overall, what was going on in the marketplace, outside of biotech, people were being very careful with capital. We were seeing a retrenching in the marketplace. Generalists were pulling out. So, it's not that we all live in a silo and the rest of the ecosystem doesn't impact us. It actually massively impacts us, and we need CEOs, senior leadership teams need to keep an eye on that and understand the vagaries of that. There's also this idea we've moved from sort of when I was doing investing initially, a Series A was like 10 million bucks. It was like 12,
Right.
15 million dollars.
And
We're
now it's
now in
50.
Series A's, right? Where it's like 50 million, 75, 150 million dollars, even up to a billion, but that's a whole
Right.
other conversation. There is I think there is a conservatism or a financial awareness that you're forced to have when you have $10 million versus when you have $100 million. And I think you build your company slightly differently. So, as we look at the access to capital that people had, I think that a lot of people took a more scattergun approach when it came to building the companies. The number of FTEs they've hired, increased substantially. The actual comp associated with that, we got people that were stepping into roles above where they actually should be. So we got title inflation. So there's a lot of corrections taking place in the market. I think for any biotech company, it really is sort of having the vision about where you want to take the company in 5 years, 10 years time, right? Being very clear about the planning from A to B to C to D, communication within the team so that you're adapting the company as data is actually coming in, being realistic from a timeline standpoint, so that when you talk about an IND date, you're very confident that that's when you're going to get there versus having to correct it because investors lose confidence in you. I think
Right.
timing to IPO. I think one of the things, and Monday morning quarterbacking is very easy to do, right? But one of the things I think we can look at is for a lot of companies that went public, they went public too early in their sort of evolution. And now we're paying the cost for that. So we're seeing enterprise values below cash on hand. So negative EVs or very limited positive EVs in there on the public side. Data not flowing at a point where it's truly catalytic and the market's going to notice it. And even arguably, the market is not receptive or is not responsive to data that is not sort of a POC in a human clinical study.
Right.
So, these companies have to survive in that backdrop, in a highly competitive system where everybody's out raising money or a large number of them out raising money, and those catalysts just aren't there. Doing it in the private market, it can be easier at times, depending on how much money you need to raise. It's not so obvious. You're not being marked to market every day. But after saying that, I think the correction in valuations on the private side has not totally taken place yet. It's still actually happening.
Yeah, that makes sense. When you're in one of these situations where maybe you've got a differentiated position and it's you and one or two others potentially going after a new technology, where does the where does the bottleneck shift to? So let's say if we take the example of Intellia and CRISPR and Editas maybe at the same time, there's a shared core technology, but you all have slightly different views on the market. But what do you do? What becomes then the arena in which you compete? Is it around the selection of the disease you go after? Is it around something like who can most effectively run trials, find patients? Because I'm just thinking about in the long run, how, there's usually one or two people that manage to really commercialize and make an impact with the technologies, and not everybody can get to an at scale company the way that Intellia and some of the others have. So I'm curious of how you run that race over the 5-10 year period as you think about it.
I'll give you a real life case today, right? So we started a company with Mike Mitchell, called Liberate Bio, and Teresa Ranke called Liberate Bio, which is focused on non-viral extrahepatic delivery of nucleic
Hmm.
acids, right? So one of the biggest issues I believe we face, of which there's probably three or four of them, but one of them is delivery. So when you look at all of these modalities we have today, they really are pretty spectacular what they can actually do. So if we think about versus 20 years ago, what we actually can do to DNA, to RNA, what we can manufacture within a cell, it's pretty spectacular. But our delivery capability is really very limited. We can get into liver, we can get into eye, we can get into spinal cord, and some other regions, the brainstem, for example. But broadly speaking, we cannot get, targeted delivery into a lot of different tissues. And for example, systemic delivery crossing the blood-brain barrier. So there's some work that obviously Denali has done with FSTAR, and I sat on the board of FSTAR as chair for a long time. I mean, there are things that we've done that is really interesting, but at the same point, we still have these limitations. So we started this company, Liberate Bio, to actually start to enable us to actually do screening of lipid nanoparticles so that we can, sorry if you can hear my dogs, we can start to get extrahepatic delivery, right, of payloads, and identify novel lipids and formulations of the LNPs themselves to enable us to do this. So when we started the company, and within Khosla Ventures, we're the only investor today, right? The intent was to let the science drive us. So get into non-human primates quickly, do the screenings, identify where these lipid nanoparticles were going, and let that then effectively direct us as to the right
Right.
indication or the right application. So, 2 years into this company, they've done a great job in getting the system up and going, called Raptor platform. We've identified now multiple lipid nanoparticles that will allow us to target extrahepatically. And the team sat down and took that data and then said, okay, so how do we deploy this? Like, what is the most relevant indication to go for where we are uniquely positioned to enable us to do that? And we've ended up in a space that they haven't disclosed yet, but we've ended up in a space that really actually has enabled them to do that. So the bottleneck, I think, along the way was going to be, what does the pipeline for the company ultimately look like? And we had 2 years really of not having visibility on that. We collected the data and now we have visibility on it. And I think that now opens up a whole opportunity set for the company, both from a BD standpoint, new investors coming in, and our ability now to really enunciate the long-term vision and goal for the company in a way that other investors will actually understand. The other one I would say, again, in real time, is Everyone Medicines, right? So we're fortunate that we have a syndicate of other investors that we're working with. We're the largest investor in the company. And that really is truly personalized medicines. So, when I was looking, very early on, and I was fortunate enough to do some work also with Alnylam, it was, we have all of these technologies. We are talking about building blocks, as we think about a nucleic acid drug. In case of CRISPR-Cas9, it's a different guide sequence. In the case of an siRNA, it's a different sequence in there, but that's programmable to
Yeah.
the sequence in you that you want to hit. And then with whole genome sequencing coming all the way down, to circa $100, and we'll see it continue to fall. So it's why are we not looking at truly personalized medicine where we're sequencing you, identifying a mutation, and then actually
Yeah.
designing a drug for you? So that's what Everyone Medicines was founded to do, with Julia Vitarello and a number of others. And Julia with Tim Yu were the leaders in a personalized antisense oligonucleotide for Julia's daughter, Mila. So Amilicin was the drug that came out from it. So here we're now looking at how do we build a relationship with the regulatory bodies out there that allows us to be able to design drugs that really are N of one or N of one plus drugs, get them into patients, get them treated, track, the overall response from these patients. So the bottleneck in that situation is clearly a regulatory
Right.
bottleneck. It's not a technical, you know, bottleneck really for us. It's a regulatory and then a reimbursement as we think about paying for these drugs. So, there's no clear answer to your question because every company just has such different elements that they need to deal with before we really get that sort of catalytic event that transforms the company and allows us then to be able to say this is where the company is going and how we're actually going to build it.
Yeah, and I'd like to talk a little bit about Everyone Essence because I think it's an amazing company, and I this point you made about the regulatory barriers is a really interesting one to dwell on because I'll play it back just to make sure that I'm getting it and the audiences as well. But essentially, in a typical drug development process, you're getting a molecule approved, right? And you're showing that it's safe, you're showing that it's effective, and the regulators approve that molecule. In this N of one paradigm, every molecule is different, and so you kind of break the paradigm in a way, right? And so but you have to, I guess, work with the regulators to think a little bit differently to say if we want to treat this long tail of ultra rare cases, maybe we get a process approved or we get some other way of approaching it approved, right? Where you reach a particular safety threshold and you prove safety and effectiveness, efficacy in a smaller group of people, but then that gives you the ability to I guess have um the blessing to take that to a much larger group of people, knowing that you've explored the bounds of the technology. Is that about right? Is that the way to think about it, or is there a different way of
It
thinking
is.
about that?
I think, it is generally, right? I think there's a very basic step, right? Or basic consideration here. You have two children in front of you, right? Both of which have a disease that will likely result in their demise in the next few years. One is where they are one child of, let's say, 1,000 children out there, right? And that there's a drug already on the market. And we see that in something like cystic fibrosis. And
Yeah.
then the other child is a child that has a rare mutation either within the CFTR gene, right, for cystic fibrosis, or, for example, ataxia-telangiectasia, right, so AT, right? We have technologies to deal with both children, right? So why is one, why do we have a structure for one that we can get a drug and we can treat them and hopefully at some point move to curative therapies, and the other one actually we don't. They don't have the right to actually have access to a drug or access to a regulatory construct that would allow us to treat them. And I think fundamentally that's the question, because the technologies are there. And to your point about, when we look at it, the way I look at this is a little bit like a CAR T or where things are going with, for example, CAR NKs, right? We're having a process being approved for them, right? And as long as the release characteristics fall within a certain specification, then that drug can be released and treated, right? If you think about
Yeah.
a CAR T, for example, it's a pretty complex drug. You've got a
Mm.
lot of different cell types that are in there. You've modified them, you've grown them up, you're shipping them back,
Yeah.
so there's a lot of complexity around it. Now you can argue it's the same CAR all the time that's in there, but everybody's different, right?
Yes.
So
Yeah,
when
it's a
you
really good parallel. I didn't think
yeah,
about
when you
that.
think about a nucleic acid drug, we can talk about it being the length of it, right? We can talk about the chemical modifications to it. There's only four bases that are being put in there, right?
Right.
Plus then chemical modifications to them. So, I can turn around and say, well, actually, we can set up assays that very clearly characterize or can characterize this drug. And if we can agree on, the framework for a release spec for a drug, right? Why would we not do it here? So when we're looking at where the FDA is slowly going, they are now going into where you're utilizing the same thing, for example, a Cas9 mRNA, or a specific capsid, that once you've validated in one, as long as it fits those release specs, you can actually keep moving forward. And I think that's the way we need to be moving for sure. So that is the plan, and I genuinely see no reason today as to why we should not be able to put this in place.
Yeah.
I think that's just engaging with the regulatory bodies then, and certainly MHRA have been great around this, right? Engaging with them and basically saying, okay, well, what does that spec look like? How do you validate it so that you can ensure that you are looking at the right spec when you go more broadly in the number of, for example, ASOs? But the intent is not to just do ASOs. I think any nucleic acid-based drug, ultimately, you should be able to develop for this.
Yeah, fascinating. I love that model, and it's company I'm going to be watching for sure. You mentioned three or four issues: delivery to the right tissue, regulatory, reimbursement, probably related to that. What are the other big barriers or bottlenecks that you see?
I think, there certainly is this element of, what is the right target from a
Yeah,
point of intervention? Are we intervening at the right space? I think that the clearly preclinical models are remain and always will be a big issue for us, and it's not clear to me how we're going to actually overcome that. And then I think it's patient, segregation in the clinical studies and tracking of them. One of the nice things within KV, we do a lot of different areas. So one of the areas we look at is digital health. We look at payer systems. Um, the digital health, as we look at advances that are being made there, overlaying that into our drug discovery and development, especially in the clinical development side of it, that starts giving us a lot of data around these patients and how they're responding to drug from a quality of life standpoint
yeah,
in real time. So, there are aspects of that I think that we're starting to see modification of, but I think ultimately patient enrollment criteria, being able to stratify our patients and being able to modify our clinical trials as we're actually getting that data as it comes through. So when we think about oncology, if we go back years ago, you were told you have breast cancer, for example, or lung cancer, and there was like, okay, well, you've cancer, right? Today, now we're talking about what are the actual cellular characteristics of your cancer? How do we now actually think about developing the right therapeutic intervention for you? So I think we've done a phenomenal job with immunotherapeutics, both with CAR Ts and sort of checkpoint inhibitors. But when we're moving into solid tumors, the variability on a patient by patient basis we see there, depending on stage, depending on individual environment that they've been in, being able to take that data, analyze it, and develop a drug specifically for them, is becoming more of the path I think we ultimately will need to go. And once you develop it for one individual and you set those characteristics, you'll find that that one individual actually turns into 20 individuals, right, over time.
yeah,
So you're putting the same drug off and you off the proverbial shelf and treating them with it.
yeah.
So, but you're back into a payer system, and then I think the cost of drugs. I they're this is where I get myself in trouble, right? So like we can all sit and talk about the value proposition of a drug that is X million dollars, right? And I totally understand that. But when we're talking about you have a dollar per, just using as an example, you have a dollar per year to pay for healthcare. That dollar doesn't become $2 overnight. It's a dollar. So, how do we think about the utilization of that dollar? How do we actually deploy that dollar? How do we actually look at ways to get these drugs a lot cheaper? So, if we really want to move from small ultra orphan or orphan indications into much larger indications for these technologies, where they are actually applicable, we've got to figure out a way to actually deal with the overall cost. That's a cost of goods standpoints,
Yeah.
development time, development cost, and then overall reimbursement. So I think we have a lot of work that we actually still need to do on that front. And obviously, things that are happening with PBMs is one aspect of that. But I think we have a much larger structural issue that we need to deal with in our industry.
Yeah, I mean, is your gut that most of the work needs to happen on, I can think of a few levers. There's reducing the cost of developing drugs in the first place. There is figuring out better ways to, I guess, internalize the cost isn't fully internalized to the healthcare system, right? We have it's hard to model out what saving a child's life actually the impact is on an economy or a person or a family over a long period of time. There's also the some of the US specific things you mentioned, like PBMs that aren't such an issue in UK or other places. But how do you break down the problem, I guess? And because when I look at it, I think about if we have the technology to get cures for all 7,000 rare diseases in the next couple of decades, we certainly don't have the health system and structure and incentive alignment to pay for those kind of things. So it's definitely we're headed for something is going to have to give there.
Couldn't agree with you more. Something does need to give, and it is, um, you look at the GLP ones, excuse me, having a profound impact,
Yeah.
obviously, on obesity and cardiometabolic. And it looks like as more studies are being completed, we're seeing more applications for them. So there is an element of how do we fail faster, or how do we identify the right patient population? Can we get drugs faster to the clinic? We're working in a very antiquated regulatory structure, and I understand why. This is not finger pointing and saying they need to change it now, or that they've been slow to change. FDA have been great, in bringing new technologies to the fore, moving things into phase 4, looking at data, working with industry. But we're still moving very slowly around that.
Yeah.
So the question is there a model that would actually enable us to be able to move forward faster, get data faster, use the systems that are in place, digital health, AI, start layering them in? Not that they're going to have a profound impact today, but actually looking at a year from now, 2 years from now, setting the system up structurally so that actually we're generating that data that actually with the regulatory authorities, that then we can actually put it into the system in 2, 3 years time. And we start to see, the net impact, positive impact that should actually
Mhm.
have, both from a cost standpoint, time standpoint, patient-centric standpoint, and physician standpoint. So I think that there is an element there that we really do need to actually start to work on, and I'm not sure that that dialogue is necessarily taking place. And I look at groups like pharma, bio, I'm not. It's not clear that really they are kind of having those or engaging in those dialogues, which is absolutely critical, to the regulatory authorities. What we are seeing is groups like, the UK. We're seeing in other areas in the Middle East. We're seeing it in other parts of Europe, in Asia. That there is this actual big drive to say, okay, we have all of these new technologies, we've all of these new learnings. Like, how do we leverage them, and how do we actually now get more? How do we adapt to them and be faster in actually getting drugs to patients at a price that actually makes sense? And I think that
Yeah.
that's the element now that we kind of need to overlay here in the US, and maybe more broadly from a global standpoint.
I want to get your perspective on AI. Khosla Ventures in particular was extremely early on OpenAI and made a great investment there. And yeah, you're an operating partner there. Some people will say biotech is different. We don't have the data richness that the LLMs have. Maybe we can generate it. I'm interested in your kind of hype meter right now. Do you think that people are they underhyping the potential impact of AI on our field? Is it being overhyped? Is it about right? And within that, I'd love if you could just pick apart where are the areas where you think we're really going to see a big step change in what we do, and where are the areas that maybe we shouldn't we should pump the brakes a little bit on what's going to happen, because it's a mix, isn't it?
Yeah. So first of all, OpenAI and that investment predates me. I think that it just shows the KV team and how they think about the continued evolution, right, of technologies and groundbreaking opportunity sets that are out there. I think there's one aspect when you can look at those technologies and direct applicability in various aspects of our lives straight out of the gate, right? And that you've got an infrastructure, or you have a tech um, enabling, that can happen in real time, and people will embrace and utilize. Drug discovery and development is different. And to your point, when we think about the training sets and the training models that are out there, what we certainly have learned is that, the quality, the consistency across different data packets, let's say, is highly variable. And when you look at scraping, people will talk about going in and actually scraping data, from publications. Again, you're back to the assay that was set up, the reagents that were used, the consistency of the data, the interpretability of it, and actually equally as important is negative data, and that doesn't really get published, right?
Yeah.
So I think we have a big issue around a dearth of data to actually train these systems in. I think the second aspect we have is, I think everybody, not everybody, a lot of people believe that this would have a profound impact straight out of the gate. And this is a comment we get all the time, right, in the industry, which is, look at the impact it's having on X, Y, and Z outside of biotech. Why are we not seeing the same thing in biotech? And there's, drug discovery and development is it's hard, right? I mean, it's there's a lot of uncertainty around it. The regulatory framework just hasn't changed. So,
Yeah.
it may help on the target identification or protein, modeling, docking, novel target identification, but the regulatory framework is still there. So we still have to deal
Right.
with that. And we really don't know until you put it into a human what's actually going to happen, right? Just being very practical about it. And then I have a conversation with people about, well, what's a platform? Is one drug, a platform, or actually is it if you've shown it three, four, five times? And to me, you've got to show it at least three times for you to know you have a
Yeah.
platform that is a durable platform, right? So I think that there's an expectation about net impact in drug discovery and development that is not realistic, right? Um, so when we see, I do believe it's overhyped right now. I think valuations are starting to get out of whack in the AI biotech space today, and we have an issue, I think, structurally, where we have drug discovery and development people that really understand microbiology, understand the actual process of discovery and development, but really know nothing about LLMs, right? And then
Yeah.
you have people who really know a lot about LLMs, know how to take data and sort of process that data, but actually are not drug discovery and development people, right? And I think that there's at times what we're seeing is that there's a disconnect in communication and language around that. And one of the things I think that we've seen over the last couple of years is a bridging of that discussion and people getting that sort of cross education, right? So we're getting to a point now where the programs themselves are better, right? We're seeing more data coming through that's actionable, that is actionable data. We have people that understand both sides, you know, um, of the divide here. So I personally am very confident that we're going to see a profound impact in overall drug discovery development and longer term treatment of patients, right? But I think it's going to take us a while. And, if things like antibodies, recombinant proteins, any of the editing technologies teaches us, it's always there is this big expectation that takes place. It drops off for a while. You may see these hype forms coming, and then it hits, and there's a consistency out there. And I also think there is an understanding of where is it applicable. So I look at CRISPR-Cas9. If I look back at the original presentations all of us were making and what was being discussed generally in the marketplace, this was a Swiss Army knife that could do everything, right? It could
Right.
make you vanilla ice cream, it
Yeah.
could bake a cake for you, it could do everything, right?
Yeah.
And there was really only one CRISPR-Cas9, Cas9, I mean, that was the one. And now you fast forward and you're like, well, actually, whoa, 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 what it does is it's very good at cutting DNA, right? And now
Yeah.
we know what Cas9 looks like. We can now identify all of these other ones that are actually out there. Oh, and by the way, now with computational power that we have, we can actually start to design these, modify them, make them smaller. So I think that it's really actually understanding where is it directly applicable today, and then
Mhm.
where is it tomorrow, where is it in a year's time, where is it in 5 years' time, and I think we may be setting it up for an expectation that is inappropriate with what's available to us today.
Yeah, it's interesting, and the point that you made around that we are in a highly regulated industry is an important one because I think if we assume that isn't going to change, then just thinking about the areas you highlighted, it's probably not going to affect our understanding of how to deliver to the right place. And until we get an AI that helps make scientific breakthroughs, then the scientists are going to have to figure that out. It may be able to help with targets potentially. I don't know that we've seen a huge amount of evidence yet there, but then all the other pieces downstream, if we start talking about using it to model trajectories in clinical trials so we can reduce the size of control arms or make them more adaptive, a lot of that is really dependent on regulation accepting that and changing. So there's quite a bit of the potential impact that's probably contingent on more flexibility, maybe from regulators and how we think about keeping the promise of protecting patient safety, but while providing maybe a little bit more flexibility around how we prove the impact, right? Is that a fair summary?
Absolutely. I think the other thing too is looking at companies, the failures that companies will have will teach them. And it's an iterative process. So, writing a company off if it has a failure, I think is the wrong thing to do. The reality is we learn a lot from that data as it's coming through, and that makes these models much better. It allows us to iterate on the models. So, as we've seen with any technology, that failure, there is a certain failure rate that you're going to see early on in the technology's evolution that really then enables it to actually be very successful. So I look at siRNA, first few clinical studies, as far as I recall, weren't great. Oncology wasn't the right application for it.
Mm.
RSV wasn't the right application for it. We the same for antisense oligonucleotides, same for proteins, antibodies. So I think that people need to temper their expectations for what it can do, understanding that this is a long-term sort of evolution that will take place, but it will hit, and it will hit in a very significant way. Just when it's going to do that, I think will be multiple years out.
Yeah. I wanted to do a quick plug for your podcast and ask about it a little bit. I really like it. It's one of the ones on my regular rotation. It's called Between the Biotech Waves. If anyone's interested in hearing more from Ness, so talk a little bit about that. Why did you decide to start it? And you have some really interesting people, and you go through some of your own stories as well. I mentioned, and you mentioned the triplet therapeutics one that was not too long ago that I found to be really good. But what's the story behind your podcast?
Well, first of all, thank you for bringing it up. It's a lot of fun to do it, and I'm very thankful for all the people that have participated on it. And we've a whole new group of episodes that'll be coming out soon. Um, I think there are groups that do a great job, and your podcast is wonderful, right? That answer specific questions, perspectives on the on what's happening in the biotech world and ecosystem overall. I think one of the things that always struck me is more of the personal story about people, the individual that actually, or one of the individuals that founded these companies, what was going through their head, how do they feel about it, why did they do things in a certain way, why did it work, why did it not work. There are times we'll see, the um, media pick up certain stories around people or around companies that really are, can be at times very one-sided, and sort of getting the perspective from people as to actually what was going on within the organization at that period of time. How did it evolve? So, I've Peter Barr, for example, coming up in one of the podcasts, and listening to him talk about the sequencing the human genome and what was going on
Yeah.
around it, what was happening with Celera at the time and how things moved and how Celera itself even came to being. So a lot of that gets lost in the storytelling. And also the trials and tribulations that people face, as they're building the companies. I think people only tend to focus on the positives and think that it was very easy for them. I know
Yeah.
from my own experience, people, thought Intellia was really easy, and that it was simple to do it. And the
Right.
reality was there were some very dark days, where we were down to, weeks of cash, so we didn't know how the company was going to go. And some of the critical decisions that we needed to make that we didn't know the outcome for them. So I think it's getting more of the insight in that, and getting their thoughts around it. And then where they think things are going to go, as they think about the next company, whether they do a next company or what do they want to pass on. So that was the genesis of it. So it's a very conversation, but it's very driven about why did you become CEO of X or take this job? Walk us through your history to this point and how you built the
Yeah.
company.
As we close out here, if you're looking forward, I'm curious about one or two of the areas that you're most excited about, especially ones that might not be on as high on people's radar. Can be technology, could be a particular therapy area or a company, something that you think is worth keeping an eye on.
So, it's a very frustrating time to be in the business. I've been doing this now for 25 years, and it is, I don't think that I've finished, another year and just been like, is this
It's
the right
wearing you
business for me
down.
to be in?
Yeah.
To be perfectly honest with you, because we have these amazing tools. Like if I think about the toolbox we have available to us today from a modality
Yeah.
standpoint, it is pretty phenomenal. When I think about what's going on with AI, when I think about computational power, when I think about access to patients, the sort of deep analysis that you can do down to a single cell and the data that's coming out from that. So it's a time where I'm looking and saying we have never been at in our industry a better place than where we
Yeah.
actually are today, right? And yet, when I'm looking at the capital that's flowing in, when I'm looking at the level of interest that people have and the level of understanding that people have about what we can do, it's just not there. So,
Mhm.
it feels like pushing water uphill, where you're actually trying to get engagement and interest within these companies and actually raising capital for them. So
Does this not make it a great time to be an investor, though?
it does
Because
make
you,
a great time, but one investor cannot really bring a
yes,
company from zero all the way through, right? Just the level of capital that's required. But even putting capital aside, having each of the different perspectives around the table
yeah,
from both investors and independents is absolutely critical because you're only better for that dialogue, right?
yeah,
So building out and having the right syndicate in place. We've got a great syndicate at EOM. The conversations we have, the company is, and I am better for every one of those conversations that take place because there's a dialogue and people are questioning, and it allows us to evolve the overall strategy for the company. So it is a great time to be investing, but we're not seeing that capital sort of flow. I think we remain having too many Me Too companies in the industry that are not differentiated.
right.
And I think there's a Darwinian selection that's taking place that needs to happen. Which is good. People are refining their portfolios.
Yeah.
They're looking at the ones that they want to back. So, I think it ultimately is a good place, but it is just the last couple of years have been very, very frustrating.
Not for the faint of heart. What needs to change? Obviously, interest rates help. Biotech innovation is going to pay off way in the future. So having 5, 6% doesn't help anybody. But what else is it? Is there fundamentally cyclical that people will change in the next couple of years, or do you think something shifted fundamentally?
So I think it is cyclical, and we've been through now 3 or 4 of these cycles. So it's certainly cyclical. The problem is you just don't know when it's going to turn, right? In
Yeah,
in any
yeah.
direction. That is a problem.
Right.
So I think it's a waiting game somewhat. I think, as we look at the new administration coming in, that's going to add regardless of who it was going to be, it adds some uncertainty as we look at, healthcare overall and how things are going to move forward. The IRA added certain uncertainty within the industry that took a while to actually address.
Right.
So, pharma has also got has an issue both with patents running out. When we look at, the money that was made around COVID, so the profits that were made from that, and the net impact had on them. So, they're going to be out hunting for companies to acquire, but these are companies that are going to impact from a revenue standpoint. So we're seeing a shift from platforms to products right now, but platforms are very important. So,
Yeah.
so money's not going into that sort of basic aspect of it. So I think that there is a cyclical. Is there a fundamental shift? I personally think that there is a fundamental shift, right? And I think that fundamental shift is that as an industry as a whole, we're starting too many companies,
Mm,
and it comes back to it's almost we're back to companies that really can differentiate. If there's $100 in the venture industry to invest, and you've got 10 companies, it's a very simple view, right? You got 10
yeah.
companies, you have $10 per company, right? If you have $100 to invest and you have 1 million companies out there, that becomes more difficult for you, especially when the companies fall within very specific buckets. So it becomes more and more difficult for you to figure out which one you should put in. Should you wait until they get that data, pay more for them, but wait until you get that data? And I fundamentally think that we have that issue in the industry today, given the number of companies we have. And when we look at management teams, not everybody can be a senior leader. It's just not possible. Not everybody wants to be, right? So do we actually have the management teams that will actually enable these companies where they are positioned for success to really be successful? Because that
Yeah.
really determines, I think, whether a company is going to be successful or not. And we don't. We don't have all of those people, right? They're being trained, hopefully, a large number of them being trained right now for the next series of companies. So I think we have some fundamental issues within our industry today that that time will address, but we're just living it in real time right now.
Yeah, yeah, makes sense. Well, thank you. Thank you for your perspective on that, and also thank you for all the impact you've had over the years.
That's very kind of you.
I said 6 biotech companies, but I'm pretty sure we covered at least 10 in the episode. So
Well,
you
thank
have
you.
every right to be a little jaded about how it's going right now.
Well, thank you very much. This is great. Thank you.
Yeah, my pleasure. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah who produced the show, and James Pierce from Selected Frequencies who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.